All right, let's go ahead and get started. One quick announcement I wanted to make is next Tuesday we'll have our first assignment due. I haven't posted up on Colab yet, but I'll post that later today. The first assignment is just to identify the biological problem or disease that you want to try and focus on for your kind of semester-long project. It won't be a long assignment. This is just a matter of trying to identify, submitting a brief description of what you want to study. For those of you who stay up late at night thinking about biological questions and diseases, this is your opportunity to satisfy that curiosity. If there isn't something that you already have in mind, that's okay. One thing I might recommend is you can actually look through and peruse some of the research that's going on in the department right now, and kind of use that as a springboard to get some ideas turning and to think about what might be a question or disease that you might want to try and focus on. Are there any questions? Great question. So the specificity, I would say, is it needs to be cellular or molecular biology focused. That doesn't mean it can't be on something with respect to dieting. If you kind of came at it from an angle of cellular molecular biology, you could try and answer some unanswered question. And so the idea that we're going to try and do next after that is to identify what unknown mechanism you want to try and focus on, or what new proposed therapy you might want to try and propose. So if this is a disease with an unknown mechanism, you could try and define, try and identify, propose a new mechanism. Or if there's already a mechanism known and already therapies that are available, how might you build upon and improve those currently available therapies? Yes? You're going to be submitting the research topic, like the disease or biological problem. So like we're going to be identifying. You're going to be submitting a few sentences describing, I'm going to try and tackle Alzheimer's disease perhaps from this angle, or I might be trying to tackle fibrosis in this organ from this angle. So the follow-up will be proposing a hypothesis. So the idea is that as you're trying to look into this disease a little bit and trying to read primary research articles, you can get started on some review articles to kind of get a landscape of what's known and what's unknown. But then switching over from review articles give you the current landscape of what's known and what's unknown. Research articles are trying to answer a specific question. And so switching from review articles to research articles will give you an idea of how to propose these hypotheses for the next assignment afterwards. I thought I saw another hand. Yes? Are we able to work with each for this project, or just Great question. So you'll be working individually, but there will be points throughout the semester where I'll be setting up, for lack of a better phrase, like study sections, where people will be grouped based on the subject and be available to kind of give each other feedback. And so as the hope is that as you all spend time trying to understand and learn about the disease or biological problem you're focusing on, your peers will be able to benefit from the knowledge that you're developing and help each other give feedback on improving our illustrations, our 3-minute thesis, and our final 2-page research statement. And again, the idea is that if you slowly just like dedicate a little bit of time each week towards it, this will become a really manageable project. And develop your skills and pursue a biological question that you're interested in. And so like typically the way this course is designed in the past, there's, I would give you a specific subject that you're going to be researching on, and you'd be working in groups, really large article that you'd be writing. But the way I'm trying to redesign this course is that you guys get to pick the biological problem that you're fascinated by, and using that interest to kind of serve as the enzyme to catalyze the reaction. I'm really trying hard there. I'm sorry. But really try to serve as like the catalyst to help pursue you, to help encourage you to dive a little deeper. It was a really bad scientific dad joke. I'm sorry. Are there any other questions? All right, so we will go ahead and get started. So today we'll be talking about enzymes. We spent the first lecture talking about amino acids, and we spent amino acids and proteins. The second lecture we started talking about cell membrane and some cell structures, and now we're going to be talking about certain class of proteins that are labeled as enzymes and relating it to botulism. Botulism is primarily caused by the bacterium Clostridium botulinum. It is a spore-forming bacterium. It's commonly found in it's a bacterium commonly found in soil, but it's also found in improperly stored food. It can also be a bacterium that can be found in wound beds that are not properly cleaned. There's also one particular food source that it's known to emerge in, and the clue I'll give is that the clue that I'll underline and say is that it's a spore-forming one. Honey, that's exactly right. Particularly in honey that is locally sourced or organic, and that's particularly why 1-year-olds and younger are typically not advised to consume honey. Some of the symptoms of the botulism toxin that comes from this bacterium are flaccid paralysis. That's usually, flaccid paralysis is the inability to properly contract muscles or the inability to properly contract muscles. There's also difficulty with swallowing or speaking. There's facial weakness, there's drooping eyelids, there's also dyspnea. Does anyone know what dyspnea means? I saw a whispering of the right answer. You want to say it louder? Shortness of breath, exactly. And finally, nausea, vomiting, and abdominal cramps. Particularly the dyspnea that emerges are in cases where this can become fatal. And so there's kind of a common theme if you look through some of the symptoms, and all of it kind of starts to revolve around muscle control and muscle function. And so we'll understand why that is as we kind of get to the end of the lecture to describe the mechanism of botulism poisoning. So it is botulism poisoning is typically rare. It's on the order of hundreds of cases annually in the US. And the way in which it works is primarily via the secretion of a particular toxin, the botulinum toxin. And in case botulinum sounds familiar, that toxin is used in Botox. As you can imagine, because that is a commonly used tool clinically, that they try and there's a lot of care given to using botulinum so it's not problematic. But there are cases in which botulism poisoning emerges in a result to its cosmetic use. And what's also noteworthy is that it's among the world's most deadly toxins. The LD50. Does anyone want to define for us what LD50 stands for? Close. You're almost there. Yes. Yeah. So if you have like some population you're working with, it's the dose at which 50% die, and the LD50 for botulism poisoning is 100 nanograms. To give you, for an average sized person, to give you context for that, I wanted to try and look at other toxins for context. This is 4,000 times stronger than cyanide. So like that's 100 nanograms per, the average person was like 100 kilogram person. So that was 0.00001 nanograms per kilogram. The same LD50 for cyanide was 4 nanograms per kilogram. And so this is 4,000 times deadlier than cyanide. And the reason why we're going to use this as the clinical backdrop for our enzyme lecture is that the toxin itself is an enzyme. So we're going to now pivot to the enzyme content of the lecture. As the pre-lecture defined, enzymes are a certain class of proteins, and they're a class of proteins that catalyze reactions. They lower the threshold for a certain reaction to occur. And the way in which we model the kinetics of these enzymes is using the Michaelis-Menten model. what I want to try and underline is that this is a model. It is not, and as with all models, it's not perfect at describing everything. And there are typically assumptions that underline every model. The way the Michaelis-Menten model works is that it typically tries to show you what the saturation rate is for an enzymatic reaction when the substrate concentrations are particularly high. And it also corresponds to a specific equation as well. And we're going to try and define some of the pieces of this equation. So we have, and to also note the plot to the left, or to the right, forgive me. The x-axis, whenever you're showing a enzymatic reaction curve, the x-axis is always going to be substrate concentration, and the y-axis is going to be the velocity of that reaction, the speed. So that'll be reactions. You can think of it as the units for that are typically molarity per unit of time. We can also think of it as molarity with reactions per unit of time. And so two key pieces of the equation that's part of this model are the Vmax and the Km. The Vmax is the rate at which saturation for the enzyme occurs in moles per unit time. So it's, to come over here for a second. So this is the point at which saturation occurs. And so this speed at which the saturation occurs, that we identify that as the Vmax of that enzyme. And we identify the Michaelis constant Km where we get half maximal velocity. And so typically we identify where Vmax is and drop down to the substrate concentration for that. And that's our Michaelis constant, or Km. A couple of things to note is that the assumptions that are part of this model are two of them. The first is that the enzyme substrate complex, that's when the enzyme meets its target. The substrate is what an enzyme is going to react with. And so when they come together, the assumption we have is that the enzyme substrate complex will reach a steady state. That's that saturation point, which means over time, the rate at which that occurs is going to be zero. It's going to reach a steady state. The second assumption is that the concentration of the substrate, again, that's what the enzyme is targeting, far exceeds the concentration for the enzyme. And so with those two key assumptions, the Michaelis-Menten formula is valid or usable. So to quickly test our understanding, we are going to do an enzyme rate experiment at our desks. And so we typically get a few of these. We get these numbers experimentally. And so given these numbers that are defined experimentally of the substrate concentrations, for the reaction rates at specific concentrations of the substrate, we want to try and approximately figure out what the Km here is. And again, if you go back to the previous slide, Km is defined as what is 1/2 of the Vmax. The Vmax is the rate at which it reaches steady state, at which the reaction rate is no longer changing. So take a minute to try and see if you can figure out what the Km here is. You can take time to plot it out on a piece of paper, but just to give you a hint, you actually don't need to plot it out. You can actually look at the values to figure it out. But take a minute, you can confer with your neighbor to figure out what is the Km here. And again, it doesn't need to be exact. This is an approximation. All right. Does anyone want to jump in and tell us what they think the Km is here? Maximus? So that is the 1/2 Vmax. What's the Km that so we have the 1/2 Vmax. What is the Km is the concentration? Yes, exactly. So it's like I said, I was thinking 1 to 1.5, exactly. So the Km equals 1/2 Vmax, but it's the substrate concentration. So we see that it appears to saturate out at about 12 nanomolar per second. 1/2 of that we go up, and we see that it appears the substrate concentration that corresponds to about 1/2 of it is a little over 1. So thinking back to the pre-lecture, if we look at these this energy curve as it relates to going from a reactant to a product, which one of these illustrated reactions do you think will go faster? Does anyone want to jump in here? Yes. Yeah, correct. So do you want to explain why that is? That's exactly right. Just to try and repeat it for the sake of the recording here. B will go faster because there's less energy required to go over this degree of energy, this difference of energy, as opposed to A, which has a larger hump. And I like the way you said it. The energy at which to get over the hump. You can kind of imagine you're pushing a boulder. Which one is easier to go get to the other side? That's kind of like at least that's how I envisioned it when I was first learning this. And so it's B because the activation energy is lower. The threshold is lower to go from reactant to product here. So there are many drugs that we see clinically, whether drugs like Cipro or anti-HB drugs, are enzyme inhibitors. And so we get two major kinds of inhibitors. We have competitive inhibitors and non-competitive inhibitors. The way competitive inhibitors work is with every enzyme, there is an active site where the substrate binds to the enzyme. Competitive after the substrate binds to the enzyme, we get the products. What competitive inhibitors do is they bind to the active site of the enzyme, directly competing with substrates for that active site or for that catalytic site. If you hear active site or catalytic site, they're both interchangeable. So again, competitive inhibitors work by binding to the active site of the enzyme. Now, the Vmax is a quality of the enzyme. It is a characteristic of the enzyme. Competitive inhibitors do not change the Vmax. They're not changing the characteristic of the enzyme. But what they're doing is they're changing the Km because they're directly competing with substrate to get access to that active site. So what that means is again, I'll repeat this, that they're not changing a quality or characteristic of the enzyme. Vmax is a quality of the enzyme. So the idea is that Vmax should not change. So if you begin to push the substrate concentration high enough in the presence of a competitive inhibitor, what do you think will happen? Think about I'll rephrase it differently. Think about what will happen at low concentrations of substrate concentration, and then what will happen as you increase it and push it to higher concentrations. Sorry, can you say that a little louder? The inhibitor? Exactly right. So I've tried to crudely draw out the reaction curve in the absence or presence of a competitive inhibitor, and I hope everyone can see this. If you can't see this, raise your hand. Is it because I'm in the way? Can you see it now? I feel like the rising guy. Can you hear me now? So in blue, we have the reaction curve with with no competitive inhibitor. But what ends up happening is, again, Vmax is a quality of the enzyme. So in the presence of a competitive inhibitor, it will slow down the reaction. But eventually, as you push the concentration high enough, we still get to Vmax. It just gets there slower. And so what ends up happening is to get to 1/2 Vmax. So if we say right here is 1/2 Vmax, I don't know if that's straight enough, but we get two different Km values. And so you could think this is Km and Km prime being in the presence of the inhibitor. So again, Vmax doesn't change. That is the quality of the enzyme. The competitive inhibitor does not change the enzyme itself. And so what it ends up doing is it ends up just delaying the time in which the reaction gets to Vmax. So that'll change the Km of this reaction, but it's not changing the enzyme. Are there any questions there? Go ahead, yeah. So it's not, so these are specifically for the inhibitor. So it's distinct from the substrate itself. We have the enzyme, and I can go back over here. I feel like for me, I'm a visual learner, so I feel like having this works. And so we have the blue triangle, which is a substrate. That is what the enzyme is targeting. And so the enzyme targets blue triangle, and at the end of the reaction, you get this product. The competitive inhibitor is maroon pentagram. And so it goes in and it competes for that active site. So the inhibitor and the substrate are two different things, two different entities here. And so the competitive inhibitor is just competing for the active site. Are there other questions? Yes.
2 questions. So with competitive inhibitors, does Km go up since the curve is going down, or does it down?
So take a second look at the graph. What do you think?
So the slope goes down. So
slope goes down, yes.
So I think Km goes down, but it's also in the denominator.
So slope goes down, but Km, if you look at it, it goes up. So because it takes longer for it to get to that 1/2 Vmax, or not longer. It requires more concentration, more substrate to hit that 1/2 Vmax. Does that make sense?
So Km goes up.
Correct.
Second question is, why won't the inhibitors stay attached to the side?
Why don't they stay attached indefinitely? I think there are some, so that's a good question. There are some inhibitors. Actually, I'm gonna look that up and get back to you. I don't know the answer off the top of my head. That's a good question. Are there any other questions? Yes. Yes. And in this example, yes, exactly. If there was enough concentration of the inhibitor. Yeah. Yes. Yes, that's true. Yeah. Are there any other questions? All right. So we have competitive inhibitors, and then we have our non-competitive inhibitors. The non-competitive inhibitors will actually bind not to the active site or the catalytic site. They'll actually bind to another site called the allosteric site. So the allosteric site, or allostery in general, means working from a distance. What that means in this context is that it's actually not going to be binding to the active site itself, but it'll be binding to another part of the enzyme. We can call, so that either is called the allosteric site or the regulatory site. And now what it's going to do is it's actually going to change, the conformation of the enzyme, and thus change the conformation of the active site. And it's going to become the association with which it's going to be binding to its substrate is going to go down. And so in this example, we have this enzyme that's going to be binding to blue triangle and releasing, creating the products. But now we have, I'm going to take one step back real quick. But now we have this non-competitive inhibitor that will bind to the allosteric site and change the conformation of the active site. Typically, these non-competitive inhibitors, what they're doing is they're changing the charge, or hydrophobicity, which will then change the structure of the active site. And so there's, and what's going to end up happening is now there's going to be a lower affinity for the substrate itself. And so, again, this is now changing the This is now going to be changing the conformation of the active site, thus changing the nature of the enzyme itself. So non-competitive inhibitors will actually be promoting, in the presence of a non-competitive inhibitor, will actually get a lower Vmax, because again, it's changing a quality or a feature of the enzyme. And so even if you push the substrate concentration high enough, we're going to hit a saturation point, but it's lower than it originally would have been because we're changing a quality, we're changing the active site of the enzyme itself. Yes. So I skipped over this. In a theoretically perfect non-competitive inhibitor, Vmax, it only affects Vmax and wouldn't affect Km, but in reality, Km is altered. In reality, Km typically goes up. Yes. So typically what will end up happening is typically what will end up happening is the enzyme will create a product as a response of the non-competitive inhibitor. And then ideally with like enough inhibitor present, it'll continue to compete. It'll continue to change enough, because we don't have like, in a real biological system, you don't just have one enzyme. You have lots of that enzyme present. And so with enough non-competitive inhibitor, it'll change the allosteric site of the enzyme. And so it'll dock, it'll release, and then more will come into dock, change the active site, release. And so the idea is that with enough inhibitor present, you'll slow down the reaction sufficiently in like a real biological system. Yes.
The activation site permanently changed.
So it is not permanently changed. I'm going to double check that, but and this kind of goes back to his question as well. I'm fairly confident that it does not permanently change the enzyme itself, but I'm going to double check that and I'll report back. Yes. We're going to get to the neurotransmitter part later on today. So, but you're yeah. Yes. So in real life, what ends up happening is that this, at low enough concentrations in the presence of that non-competitive inhibitor, what ends up happening is that it changes the slope of these reactions as it starts to approach a lower Vmax. So like in an ideal like theoretical system, it doesn't change it, but in reality, what ends up happening is like even at lower concentrations, as it approaches, as it tries to hit this this lower Vmax and lower curve, that alters the Km or the 1/2 Vmax there, because it's approaching a lower Vmax. Does that make sense? Okay. Yes.
So would theoretically non-competitive inhibitors be more effective since they don't need to compete? They can just bind to whatever enzyme and just knock out whatever substrate in there.
It's all context dependent. For the sake of time, I'm gonna keep pressing on. I'll be available afterwards to answer questions, but we got to keep pressing on. So allosteric regulation. So again, allostery means working from a distance. What we just saw on the previous slide was non-competitive inhibitors work on an allosteric site away from the active site. And this again will allosteric regulation enables the changing of the shape of the activity or the active site of the enzyme. Now, what's important to note is that it's possible that this change, we saw non-competitive inhibitors can inhibit or promote the active site to have a lower affinity for the substrate. There are situations in which it can actually enhance the interactions between the substrate and the enzyme. And so there's allosteric inhibition or allosteric enhancement. Yes. You know, I haven't heard of that, but that makes sense that it would the conformation change would enable the enzyme to bind to something different. So I don't know of that, but that makes sense. Sorry, I just need a second to get my bearings. And so typically, the way in which this looks biologically is not like the illustration on the previous slide, but rather there's typically there's two different domains for this enzyme protein. We have C denotes the catalytic domain. And again, catalytic site and active site are mean the same thing here. So the catalytic domain of the enzyme has the active site, and the regulatory domain has the allosteric site. And so to take a moment to talk about domains, we've talked about proteins with subunits in previous lecture, and now we're talking about proteins with domains. And so it'll help to try and define what domains and subunits are here. So domains are distinct regions of one single protein. So typically here we're talking about a catalytic domain or a regulatory domain. But when you hear domain, that just means separate parts of one protein, of one single polypeptide. Subunits typically implies separate proteins coming together. Okay, so that means there are two different distinct proteins that have now come together to that are working as a combined assembly for a specific function. And so, as we talked about in the first lecture, we had hemoglobin, where there are 4 subunits coming together. And the reason why we're defining subunits versus domains here is that the botulinum toxin has 3 different domains. It has, and each domain, it's part of one protein, but each domain have distinct functional units. We have the receptor binding domain, we have the translocation domain, and we have the catalytic domain. Above it has, we have light chain and heavy chain defined. That is something that was defined when botulinum toxin was first discovered. The way we typically, one of the ways in which we try and assess the presence of proteins is through a Western blot where we degrade proteins. And what happened here was botulinum toxin had 2 distinct chains that emerged. And so it was first defined as light chain and heavy chain. But now that we know a lot more about botulinum toxin, the best way to distinguish the different regions is by its functional domains. So with allosteric regulation, there's a few different ways in which this can happen. Yes. No, because they don't come together for, they don't work together for a specific function, like working as an assembly together, but it's actually just the two of them work to, what ends up happening is that the two of them interact during a reaction that yields some product. So they're not working in concert together as an assembly. There we go. So among the allosteric regulations, we have covalent modifications that can emerge, where we have the addition or removal of certain groups. One example of this are kinases. So kinases add phosphate groups to other proteins. And so what this ends up looking like is X kinase comes over and phosphorylates, adding a phosphate group to enzyme X on a regulatory site, on an allosteric site. And this enables, this induces a conformational change. So whenever there's like some charged group or something added to an enzyme or to a protein in general, if you see something, phosphate groups are typically negatively charged. So something that changes the charge of the protein is going to change its structure, and that will in turn have an effect on its function. And so typically what will happen here is that this phosphorylated enzyme will now have, will undergo a conformational change, which will open up an active site that enables it to catalyze a reaction. Typically, phosphates are added to 3 specific amino acids, serine, threonine, or tyrosine. And tyrosines are typically tyrosine phosphorylation is less common than the others, but when it does happen, it's mediated specifically by a tyrosine kinase. And so again, whenever you hear kinase, that is the addition of a phosphate group, and we call that addition of a phosphate group phosphorylation. There are also a class of enzymes that remove phosphate groups, and we call those phosphatases. There is another modification where we have the cleavage of specific bonds. And so whenever you see a protein that has, typically if it has pro on the front of it, that means it's in its precursor form, or it's like not quite active form yet. And so what ends up happening here is that proenzymes are cleaved to become enzymes. And so an example of this is prothrombin in the clotting cascade cleaved to become thrombin to further enable the clotting cascade to occur. Another example is proteins binding together and associating with one another that thereby activates enzymatic activity of one of them. And so usually what will sometimes happen is when receptors come together and dimerize, when a receptor binds to its ligand, that it's binding to a ligand, that'll change its conformation because that ligand is going to carry with it a charge, and that is going to end up also changing a domain inside of it that typically has a tyrosine kinase on the end. And so that kinase will then enable phosphorylation and downstream signaling when receptors bind to their ligands. And so the take home on that association and dissociation of subunits is that when they come together, they can enable the activity of an enzyme that is a subunit within that receptor. And finally, cooperativity. What this means is whenever an enzyme meets its substrate, they form an enzyme substrate complex, and that produces a product. And then usually, because it's now a different, it's no longer the substrate, its target, it's now a product, something different, it's now going to dissociate and look for more substrate. Cooperativity is where the product now helps and affects this enzymatic reaction and further speeds up the reaction, where the product is now participating in this reaction. And that typically can imply that there's multiple catalytic subunits. And so the way in which this affects or is informing the Michaelis-Menten model is that typically the product that can then act back on the enzymatic reaction, we add this exponential coefficient called the Hill coefficient that's defined experimentally. And what ends up happening is, as you can see, as the Hill coefficient goes up, this speeds up the rate of the reaction over time. One example of this biologically involves a enzyme called glycogen synthase kinase 3, or GSK3. So what ends up happening here is that its substrate is glycogen synthase. And the substrate, in this case, is a specific motif or set of amino acid sequences where whenever it sees a phosphorylated serine, the X's denote any amino acid. So phosphoserine, any 3 amino acids, and then a serine, that is this enzyme's target. And now, again, this is a kinase. So does anyone remember what a kinase does specifically? Yeah, phosphorylates. And so what's going to end up happening is there's going to be another kinase that phosphorylates this first serine. GSK3 is going to see phosphoserine, 3 amino acids in this serine, so it phosphorylates this one. But now this product has now become its target because after it phosphorylated this serine, there's another substrate motif for that same enzyme that can then phosphorylate this serine, and that happens again for this serine, and that happens again for this serine. And so this is an example in which the product of the reaction feeds back and can increase the rate of the reaction, and then in this case, it becomes the substrate all over again. And we call that first kinase, that first phosphorylation event, the priming phosphorylation. Before I move on, I want to hit pause there. I'm going to drink some water, and I want a couple questions. Yes. What's important for you to know is that cooperativity is represented. So it's important for you to know the Michaelis-Menten equation. Know that equation. What's important to know here, though, is that the enzymes that display cooperativity are represented by that exponent, that Hill's coefficient. And so I'm not going to require you to be able to write out and memorize exactly, though I know all of you could. I'm not going to ask you to memorize what the formula will look like with the Hill's coefficient, but rather what the Hill coefficient means. Does that make sense? I think I saw your hand up first. I'm gonna jump to you next. You got it.
They're all like each one is phosphorylated and therefore the reaction.
Yeah, because if you imagine there being a signal that represents each time this reaction happens, and also we have a lot of substrate within a tube or within a jar. If there was only one phosphorylation event between GSK3 and glycogen synthase, it would require, you'd get one event per glycogen synthase. But here we're getting multiple events. And so the reaction is occurring much faster because it's able to have a reaction multiple times with this one molecule of glycogen synthase, where the product feeds back into the reaction because it can then become the substrate all over again. Yes. Yes. Yeah. Whenever you see lowercase p in front of a serine, threonine, or tyrosine, that typically denotes a phosphorylated version of it. Yes.
I'm just where all the graph and everything is that
No, because what ends up happening here, well, sorry, forgive me. Vmax is a quality of the enzyme. And so that would be, I actually think there's a specific name when there's like an intersection between different curves like that, but I can't remember the name of it, but that's not specifically, in this case, that's not the Km there. Yes. Yes. Yeah. So your question is, even though CK2 is the primary phosphorylation event here, after GSK3 phosphorylates here, does it become the priming phosphorylation event for further events? Yes, exactly right. Yes. So in this case, it can imply, not always the case when cooperativity is at play. So that would relate back to the catalytic subunit relates to the enzyme, not the substrate. In this case, glycogen synthase is the substrate. And so in this case, it's not denoting, what we're talking about here is not denoting multiple catalytic subunits, but rather that there are multiple targets on which the catalytic subunit can interact with on its substrate. All right. So for the next slide, I'm hoping the audio works. So this relates back to someone asking a question about neurotransmitters, I think, and how some of this relates back. And so because I'm not a neuroscientist, I'm going to allow this video to kind of set the stage and set the context for neurotransmission. That'll be really important for how botulism toxin works.
Communication between two neurons begins when an electrical impulse called an action potential travels along the axon of a presynaptic neuron toward the axon terminal. The action potential cannot cross the synaptic space. When it reaches the axon terminal, it causes membranous sacs called vesicles to move toward the membrane of the axon terminal. The membrane of the vesicle fuses with the membrane of the axon terminal, enabling the vesicle to release its contents into the synaptic space. The molecules released from the vesicles are chemicals called neurotransmitters. They drift across the synaptic space and bind to special proteins called receptors on the postsynaptic neuron. The binding of a neurotransmitter to its receptor can trigger an action potential in the postsynaptic neuron. That electrical signal then moves toward the cell body of the postsynaptic neuron. Now that the neurotransmitter has relayed its message, it releases from the receptor into the synaptic space. Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time an action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
All right. So the role of vesicles either in neurotransmission or also in exocytosis, when protein contents or biomolecule contents are released from a cell, is involved with a vesicle fusing with the plasma membrane, releasing its contents extracellularly. Excuse me. As you can imagine, this is a high energy event because there's an incredible amount of hydrophobic interactions that have to be overcome. And so this can't just happen passively. There's a class of proteins that enable the squeezing out of every single water molecule to enable the fusion of this vesicle with the cell membrane to enable the releasing of the contents. And what vesicles really look like are littered with all of these proteins on the surface. And in particular, the one that we're going to be interested in is called synaptobrevin. The way in which this fusion and release of contents work by vesicles is that these vesicles will fuse with these membrane proteins called snares. And both the vesicle and the cell membrane have different species or isotypes of these snare proteins. The vesicles have V snares, and the target, in this case, the cell membrane, carry T snares. And so what ends up happening is that the V snares and the T snares, as the vesicle gets closer to the cell membrane, what will end up happening is that the V snares and the T snares will start wrapping around each other, and this coil-coil interaction starts to occur, where if you can envision like a long, thick rope, like a tug-of-war thick rope, if you start to twist it and start to coil it up, what will end up happening is that you have these coils that kind of emerge in the middle. And that's kind of similar to what happens here between V snares and T snares, where they start to spin and coil around each other, and they start to pull and overcome the high energy that's required to kind of push water molecules out of the way and to fuse the vesicle membrane with the cell membrane and enable vesicles to release down out of the cell. What's important to note here is that synaptobrevin is a V snare. That means it is a snare protein on the vesicle. And the target of botulinum toxin is that it cuts synaptobrevin. So as I mentioned before, there's multiple ways in which the botulinum toxin can present within a person. One of the key ways to note is that when it enters through the gut, what will end up happening is that the botulinum toxin botulism bacterium will not be able to get across the tight junctions present across the epithelial lining within our gut. And bless you. One thing to note is that a lot of the things that we talk about in this class, and I think I kind of illustrated this or talked about this in the first lecture, some of these things are still being explored and better understood with research over time. And so there was a study, I think it was about a couple of years ago, that identified a specific kind of intestinal epithelial cell called an M cell that has less of a mucus lining. And what ends up happening is that because it has less of a mucus lining there, the botulinum toxin will then cross the epithelial layer through those M cells. And what will end up happening there is that over time, as it crosses the epithelium and gets into our bloodstream, the botulinum toxin will find its way to a motor neuron. I bet all of you are wondering, how does it specifically find a motor neuron? It's internalized by endocytosis by the motor neuron. The way in which it finds specifically motor neurons is by that receptor binding domain. So that receptor binding domain specifically targets motor neurons. And so once the toxin binds via the receptor binding domain, it'll then use the translocation domain it'll then be endocytosed. And then when it's endocytosed within the endosome, the pH change within the endosome, again, when proteins experience a pH change, it's going to change its structure, and that's going to change its function. What's really, really interesting here is that within the endosome, the translocation domain, because of this pH change, will now change its structure to basically become a harpoon and shoot its way out of the endosome before the endosome matures into a lysosome and degrades it. And so as it shoots out and breaks out of the endosome within the motor neuron, now it's within the cytoplasm, and the heavy chain and light chain will dissociate. And now we have this light chain that has the catalytic domain that will now cleave and act on synaptobrevin, the V snare. I just scooped myself and I got really excited and just said all this. So again, the toxin will escape the vesicle, and the light chain will separate from the rest. And the function basically for the receptor binding domain was to get it to the motor neuron. The function of the translocation domain was to help it escape. And now the catalytic domain and the light chain will be able to have its effect. And so the light chain, the kind of enzyme that it is called a zinc endopeptidase. And so typically there are a class of enzymes that require a cofactor, that require something to enable it to become, to enable the reaction. In this case, it's a divalent cation zinc. And the endo part of the peptidase, endo part means that it cuts in the middle. And so in this case, it's cleaving synaptobrevin. And so if there's no synaptobrevin, that's a V snare, that means the vesicle cannot fuse with the cell membrane to release its contents. And since it's a motor neuron, that means it won't be able to release neurotransmitters. And because of that, you get paralysis. So what will end up happening is that the toxin keeps catalyzing the same reaction over and over again. And the toxin is able to survive for days. And so the question becomes, how much damage can it do? How efficient is the reaction? And so we're able to kind of test our understanding here to calculate the turnover rate. And so the Vmax for a botulinum toxin is 14 micromolar per minute. And given, now we're going to do a quick question. So given that we have 100 nanomolar of the enzyme, what is the turnover rate? Does anyone remember what turnover rate means from the pre-lecture? Yeah, exactly. And so it's going to be the reactions per second. And so given what we have here, take a moment to calculate and confer with your neighbor, what is the turnover rate here for botulinum toxin given these conditions? One quick clue I'll give here is to consider that, as he said a moment ago, turnover rate is typically a per second calculation, and we're given the Vmax in per minutes. And so be mindful of the units. And yeah, I'll give you guys another 30 seconds to calculate that. All right, does anyone have an answer? Sorry? One order of magnitude off. Yeah. Do you want to say that louder? So what did you calculate for the turnover rate here? 2.3 is right. And so given that the Vmax is 14 micromolar per minute, once you you can also consider the unit being 14 micromolar of product in given reactions per minute. And so as long as you divide by the 100 nanomolar, you can convert that to 0.1 micromolar and divide by 60 to set it to seconds, you get 2.3 reactions per second. And so given that there are about 70 synaptobrevin vesicles, synaptobrevin molecules per vesicle, and given that there are about 150 vesicles per motor neuron, in a few hours, 1 molecule of botulinum toxin will be able to kill a motor neuron. So treatment. There are antitoxins that are used. They're directed against the toxin. And these will disable or neutralize the toxin that have not entered a nerve cell, but they don't do anything for any of the botulinum toxin that's already entered a motor neuron. Another route is antibiotics to try and eliminate or get rid of any of the bacterium that still hasn't, to clear out any of the bacterium. But again, that doesn't do anything for any of the enzyme that's made its way into your bloodstream or into any of your motor neurons. And respiratory support is typically meant to try and ease symptoms for patients. And so if you think bacterial toxins are cool or vesicle fusion of membranes are cool, these are a couple of folks in our research community that are plugged into those fields of research. Before I wrap up, I kind of want to give space. Are there any questions that I've left unanswered? Yes.
So it's so great, but how does this help bacteria evolutionarily? How does that
typically, so it's not that. So the way in which this helps bacterium survive, if you think about the way the toxin is designed, if you think about it having a receptor binding domain, a translocation domain, and a catalytic domain, there were probably ancestral forms of botulinum toxin where it maybe only had a catalytic domain. And then there were probably mutation events that occurred over time where it developed this translocation domain, and that enabled it to better survive and escape and carry out its function. And so it's not so much that the evolutionary constraints for botulism here are present in how each domain has emerged over time to help it carry out its function. Does that help make sense? But it doesn't do anything for the bacterium itself, though. To get to I think half of your question.
So it's kind of like something that's carried on. It doesn't harm or help. So it's
Yes. For now, I'll say that's correct.
like a stagio.
Yeah. Are there any other questions? Yes. I think that's a great question. As far as I know, it seems that there's been enough evidence shown that controlled doses delivered subcutaneously can have this cosmetic effect that, again, the plastic surgery industry is very lucrative. And so like people are willing to pay for this. and the rates at which botulism poisoning occurs is fairly small. Like again, we only get hundreds of cases a year, and not all of them are cosmetics related. And so it seems like it's been controlled to have this like desired effect. Still kind of creeps me out a little bit, but that's not to dissuade anyone from doing that in the future, but rather perhaps to be mindful of what is being used in that setting. But yeah, like I think a short answer to your question of why is that happening, people have been able to find a way to make it work without too many instances of botulism poisoning, and it's been incredibly lucrative. So I think that's, sorry. Well, like you're getting that paralysis that is getting rid of some of those wrinkles in that area, and so I didn't know that. Okay, that's good to know. For the sake of the recording, someone said that it can also be used to reduce craniofacial pain. Is that an accurate representation of what you just said? Okay. Yes. So a few ways of getting it are spores present in honey, or improperly stored food, infection in a improperly cared for wound bed, and that's typically it. Outside of cosmetic use, the two main are going to be and soil. Forgive me, that was the third I forgot. Particularly from dirty soil, food, and honey. Yeah. Are there any other questions? Yes. Yeah. So kilodalton is a way to represent molecular weight for a protein. So a Dalton is a moles per gram, and so kilodalton is like kilo for thousands, and so 50 kilodaltons is 50,000 daltons, and so then 50,000 moles per gram. I'll take by the chatter that there are no more questions. I'll stay up here for a little bit, but with that concludes the lecture. Thanks for your time.